Hey everyone, and welcome to This Explainer. Today, we're diving into the mechanics behind literally one of the most exciting AI breakthroughs of our time, AlphaFold. If you've been following computational biology at all, you already know this isn't just some tiny incremental step forward. It is a massive, game-changing leap in how we understand the very building blocks of life. We're going to break down exactly how modern artificial intelligence takes a simple sequence and predicts a wildly complex three-dimensional protein structure. Let's get into it. Okay, here's our roadmap for today. We'll start with the protein folding challenge, peek inside the AlphaFold pipeline, see how it actually learns, decode the confidence and accuracy metrics, and finally, look at its limitations and next steps. All right, kicking things off with Section 1, the protein folding challenge, specifically looking at the conformational space. To really grasp why this AI is so revolutionary, we have to understand the impossible puzzle it's solving. Proteins start out as simple, one-dimensional strings of amino acids. But for them to actually do their jobs in our bodies, like digesting food, fighting off viruses, or transporting oxygen, they have to fold into incredibly precise 3D shapes. This folding process is driven by a massive network of physical interactions. Honestly, getting from point A, the string, to point B, the functioning biological machine, has historically been one of the hardest problems in all of biology. Let's put this into perspective with a quick number. Think about a tiny 100 residue protein, which, by the way, is pretty small in the biological world. There are 99 backbone dihedrals per chain position for that protein. What does that actually mean? Well, it creates an astronomically large conformational space. The number of potential shapes that one tiny protein could theoretically fold into is so massive that if you tried to simulate every single one, the universe would literally end before you finished. Crazy, right? And this beautifully illustrates why we needed a paradigm shift. For decades, the go-to was classical molecular dynamics. We literally tried to simulate every single physical movement and atomic twitch. But as we just saw, the math is totally against us. It takes vast, overwhelming compute power, and it almost always just runs out of practical time before finding the true native shape. So AlphaFold completely flipped the script. Instead of simulating explicit physics step by step, it uses deep learning to develop learned spatial reasoning. This is what allowed it to achieve near-experimental accuracy, matching medium-resolution models during the famous CASP-14 competition. It predicts patterns instead of simulating physics. Moving right along to Section 2, inside the AlphaFold pipeline, focusing on input data fusion. Before the magic really happens, AlphaFold executes this ingenious step called data fusion. It doesn't just look at the 1D target sequence in a vacuum. It fuses that sequence with its rich evolutionary history. It pulls in multiple sequence alignments, or MSAs, from homologous sequences. Because think about it, if two parts of a protein mutate together over millions of years of evolution, it's a huge hint that they sit right next to each other in 3D space. Plus, if there are template coordinates from related, known structures already hanging out in the protein databank, it throws those in too to help constrain the fold. What's so cool here is how this complex neural network acts like a literal factory assembly line, taking raw 1D data and refining it into a polished 3D masterpiece across 4 main steps. First step, feature initialization encodes those MSAs and templates into numerical arrays. Then comes the evoformer. This is the heavy lifter. It uses iterative attention blocks to deeply capture coevolution and long-range relationships between the amino acids. Third, the structure module takes all that relational data and converts it into actual atomic coordinates using invariant point attention. And finally, there's an optional relaxation step. That's just a quick energy minimization to fix any awkward atomic clashes and give us the final beautiful coordinates. All right, Section 3, how AlphaFold learns, specifically looking at the training signals. So we know the assembly line now, but how did the AI actually learn to be so incredibly accurate in the first place? The secret sauce is that AlphaFold was trained on thousands upon thousands of known, experimentally verified structures. By intensely studying this diverse dataset alongside their evolutionary histories, it developed a deep learned spatial reasoning. It basically figured out the underlying rules of how proteins fold without ever opening a physics textbook. But, to get there, it needed some really strict guidance during its training phase. While it was training, the AI was constantly making predictions, and when it made mistakes, it got corrected through mathematical signals. One super important correction method was histogram loss. This training signal strictly supervised the distances between every pair of residues. It basically nudged the AI, saying, hey, your predicted geometry doesn't match the ground truth data here. Try again. This ensured the distances between all parts of the protein matched reality perfectly. Getting the distances right is great, but the atoms also have to be oriented correctly. That's where frame-aligned point error, or FAPE loss, comes in. This signal heavily penalizes the model if it misplaced atoms within their local residue frames. Think of FAPE loss as a rigorous, unforgiving teacher, forcing the model to be incredibly precise right down to the atomic level. Now, this is where it gets really wild. The creators didn't just want an AI that spits out answers. They wanted an AI that actually knows when it might be wrong. So they built in auxiliary heads. These are specialized components trained to let the AI essentially grade its own homework. It learned to estimate its own confidence and uncertainty, generating vital metrics known as PLGD and PAE. And trust me, these are absolutely essential if you're going to use these models. Which brings us perfectly to Section 4, decoding confidence and accuracy. Basically, how we can trust the AI. If you're using this tool in your research, this little confidence cheat sheet is going to be your best friend. It shows exactly what the per residue confidence score, the PLDDT, means for your hypotheses. If the AI gives a region a score over 90, you're looking at very high confidence. You can likely trust those sidechains. If it sits between 70 and 90, it's a cautious reliability signal. It supports backbone-level usefulness. But if it drops below 50, you are dealing with highly unreliable predictions. Actually, I find this part incredibly fascinating. A feeling grade from the AI gives us a brilliant biological clue. When you see a PLDDT score under 50, it doesn't necessarily mean the AI was just confused or broke down. More often than not, it points to an intrinsically disordered region of the protein itself. It's literally telling you that this specific segment is naturally floppy and unstructured in the real world, which is hugely valuable information on its own. Now, keep in mind, PLDDT only grades individual local parts. To get the big picture, you absolutely must use the PAE plots, or predicted aligned error. PA assesses how confidently whole domains and interfaces sit relative to one another. For example, you might have two domains with great PLDDT scores, meaning they're folded perfectly, but a terrible PAE score between them means the AI has absolutely no idea how those two domains orient towards each other in 3D space. You always gotta check both metrics. Finally, Section 5, limitations and next steps. Let's look beyond just the prediction. As miraculous as this tool is, it's vital to remember it isn't perfect. Standard AlphaFold really struggles with multi-chain complexes. If you've got multiple proteins interacting, you absolutely have to use this specialized AlphaFold multimer version instead. It also generally fails at completely modeling ligands and cofactors, which we know are often crucial for a protein's actual function. And, like we just covered, it isn't going to give you a single static structure for those intrinsically disordered regions. So what's the core takeaway here? You have to be a smart consumer of this data. Let those high-confidence AI predictions generate momentum for you. Use them to drive your immediate hypothesis generation, like planning out your next mutagenesis experiments. But treat the low-confidence regions as entirely tentative. For those uncertain bits, you really must seek out and rely on real-world physical testing. Leaving you with this thought as we wrap up this explainer. AlphaFold is an undeniably historic breakthrough, but it is not a replacement for the laboratory. It's a powerful, accelerating compass. The true magic is going to happen when we blend these high-speed AI predictions with precise experimental data, like cryo-EM. So the question is, how will you use this incredible tool to navigate and unlock the future of biology? Thanks for hanging out with me on this explainer, and keep exploring.